Yeah. Bueno, pues muy buenas tardes. Estamos en el ciclo de conferencias de la licenciatura en Ecología de la Escuela Nacional de Estudios Superiores, Universidad Nacional Autónoma de México. Y hoy pues tenemos el gran gusto de tener con nosotros a Susan Kalish de la Universidad de Tennessee. Ella es directora del Departamento de Ecología y Biología Evolutiva. Y para nosotros es un gran gusto pues que haya aceptado y que está acá con nosotros. Es una gran investigadora y bueno, es amiga también, conocida muchos años y eso es algo muy grato tenerla acá. Entonces, pues vamos a dar inicio. Thank you very much, Susan, for being here. We're really glad that you accepted our invitation. And I know that our students will be really glad too after they hear your talk and learn a lot. I think this is something that we are trying to do every week and inviting people from overseas and also from, Mexico. So we're really happy that you're here. Thank you very much.
I'm going to make a brief introduction about you. So, buenos días, jóvenes, estudiantes, profesores, investigadores que nos acompañan hoy. Bueno, como ya lo dijo Mauricio, tenemos el gusto de tener aquí a la doctora Susan Kalish, que ha sido designada como profesora de excelencia y es la directora del Departamento de Ecología y Biología Evolutiva de la Universidad de Tennessee. Hizo su doctorado, su maestría y doctorado en la Universidad de Chicago y ha tenido una larga y exitosa carrera académica en el campo de la ecología, la evolución y la conservación de plantas y con contribuciones muy importantes al entendimiento de esta relación entre la ecología de las interacciones, la genética y la evolución de los sistemas reproductivos de plantas. Todos estos temas muy importantes para nuestra audiencia de jóvenes ecólogos y nuestros invitados interesados en este tema y en la biología de las plantas. La doctora Kalish tiene más de 100 publicaciones, más de 9.500 citas a sus trabajos y actualmente en su laboratorio se abordan una amplia gama de temas, incluyendo la evolución de la autopolinización, ecología de polinización, barreras de aislamiento reproductivo, enemigos naturales y mutualistas, genética del desarrollo floral y de los sistemas de apareamiento. Y bueno, de esto último es de lo que nos va a hablar el día de hoy. Así es que, bienvenida, welcome, Susan. Dr. Kalish, and we thank you so much for accepting this invitation and dedicating this time to us. Thank you.
Thank you very much for the introduction and thank you for the invitation. I wish we were all in person to enjoy interacting and conversation, and maybe that can happen sometime in the future, but I'm very happy to be here even by Zoom. And I'm in Pennsylvania right now, which is one of my field sites. So I'm not even in Knoxville, Tennessee. I'm different from that. So anyway, thank you so much. And should I just start my talk now? Okay.
Go ahead.
So can you see it?
Yes.
Yes.
Okay, great. So I work since I was a graduate student on the mating system evolution in flowering plants. And I want to start my talk by, oops, come on, let's see, there we go. By thanking a number of collaborators who have worked with me, and I've worked with them over the years, who have really helped to put this system together and to give it the wheels that it has now. I've been funded by a lot of different agencies and universities, including the University of Pittsburgh, the University of Tennessee, the National Science Foundation, different working groups from the Micromorph Working Group and the Nescent Working Groups on Mating System, and the Botanical Society of America and PEER, which is a University of Tennessee support for graduate students. So when you think about how evolution drives mating system evolution, what forces are working in natural populations, there are genetic forces which involve primarily the transmission advantage, that is passing on more alleles or more gametes through selfing. But this is counteracted genetically by the expression of inbreeding depression, which is the lower fitness of self-progeny relative to outcross progeny. And the ecological forces that are involved in the evolution of selfing include outcross pollen limitation, pollen discounting, and increased fitness under suboptimal conditions. And I'll be talking about these different forces in the different parts of my talk. These forces act on any organisms that are hermaphroditic. So those can be hermaphroditic fish, snails, slugs, corals, these sorts of things. And they have a profound impact on the mating systems and inbreeding of many marine organisms. But when we think about mating systems, those of us who are plant biologists and botanists, we think about hermaphrodites where the flowers of the plant have both male and female parts within the same flower, and the likelihood of self-pollination is not zero. So there's an opportunity for the evolution of selfing to occur. When you look at the distribution of outcrossing rates, that is when people measure how selfed or outcrossed the progeny of individual plants in a population are, and then average that across the entire population, we see that for abiotically pollinated groups like wind pollination, there are sulphurs and then there are outcrossers. But for biotic pollination, that is plants that require require a pollinator of some kind, a bird, a bee, a bat, to move pollen, we see that there isn't this same break, but there's a continuous distribution across plant mating systems. And this suggests to me and to many others that mating system itself as a trait of plant populations and species is dynamic. So this is a meta-analysis by Whitehead et al. From 2028, looking at 105 different species, and this value on the y-axis is the population outcrossing rate, and individuals up here are very high selfing, species down here are high outcrossing, populations down here are high selfing, and those in the middle are somewhere between outcrossing and selfing. And you can see if you follow any of one of these lines up and down, that within a single species, there can be tremendous variation among populations or among years in the estimates of outcrossing rate. And so this suggests that mating system itself is very evolutionary dynamic and can move between high rates of selfing and high rates of outcrossing. So during my career, I've been working to develop Collinsia and Tenella as model genera for studying the evolution of mating system. And this group is particularly interesting because it has multiple species pairs where one individual species in the pair is large flowering and primarily outcrossing, and its sister species is very small and primarily selfing. And this is repeated over and over, so over and over, over and over, over and over within the genus. In fact, there 7 or 8 examples where the closest sister species to an outcrosser is a sulphur. Now, Collinsia is widespread in northern North America. It goes into Canada. It's all along the west coast. Some small flowered species go across the Rocky Mountains and into eastern North America. And there are some species, grandiflora and parviflora, that are in northwestern Mexico. And what we've seen in this group is that the outcrossers have these large showy flowers. They look obviously like outcrossers, and their sister species have evolved small flowers and are kind of diagnostic of what we think of as the selfing syndrome, and where there's reduced floral display, lower pollen, and higher rates of selfing. And in fact, I just put this in today that in the Wikipedia, under the selfing syndrome, the species pair grandiflora and parviflora are the picture children for outcrossing and selfing syndrome evolution. And so I feel like we've made it as a model system somehow, finally. So what I'm going to talk to you about today is different approaches that we've taken to look at how evolution of selfing or outcrossing evolves or doesn't evolve. And so I'm going to talk to you about 3 different parts of work that we've done over the years. And I'm going to start with how sympatry can influence the evolution of selfing. So this is work that was done with April Randall and Rachel Spigler. And so when you think about plants in sympatry or congeners in sympatry, so that they're co-occurring, you know that individuals that are congeners will be very similar for a number of traits, including floral traits. And they will likely express competition for resources, especially pollinators, which can reduce seed set and can increase selfing. So there can be divergence in traits related to shared resources. So you'd expect the populations to diverge in these, or the co-occurring species to diverge in mating system traits. In addition, heterospecific pollen transfer, that is transfer between different species in the same genus or other genera, can cause loss of pollen, stigma clogging, and if the species are related to each other, can cause the production of unfit hybrids, which will drive the evolution of reproductive isolating barriers. So reproductive isolating barriers are of 2 kinds, pre-mating, which are physical separation, behavioral separation, habitat preference differences, pollinator preferences, or flowering time shifts, and post-mating, which is prezygotic and post-zygotic factors that cause hybrid instability or inviability. So we were interested in understanding whether or not pre and post-zygotic traits or factors were driving the expression of mating system. And we worked in Oregon, which is in the Western United States, on C. linearis, Collinsia linearis, which is large and outcrossing, C. rutanii, which is small and primarily selfing, and in areas where both species co-occur, shown by these half-filled circles. What we did was look at the overlap in flowering time to see whether the possibility for gene flow could exist and hybridization could exist. And we found that in all the sites that we looked at here, 4, where they co-occur, that they basically overlap. And so the opportunity for hybridization is really high in these sympatric populations. In the populations where they were sympatric, we also looked at the pollinator movement rate between the species. So what we saw is that when the populations were allopatric, so the gray bars are allopatric, that is, they're living by themselves. Ratanii gets about the same amount of pollination as Linearis, but when Linearis and Ratanii are together, there is more visitation to Linearis, but roughly about 15% of the pollinator movement is between the two species. So there's potential for visitation across the species. So what does that mean? Well, we looked at the cost of hybridization that could be paid by Ratanii or Linearis. And what we found is that so this graph shows the proportion of seeds that germinated when Linearis was the female parent, the dam, versus Ratanii was the female parent, and receiving either Ratanii or Linearis pollen as the sire. And what we see is two things. First of all, Ratanii is terrible at pollinating Linearis. It's really bad, and it's significantly less than Linearis pollinating itself. But when you look at Ratanii pollinating itself or Linearis pollinating Ratanii, there's no difference here. And in fact, Linearis is just as good at pollinating Ratanii as Ratanii is pollinating itself. So this means that Ratanii can potentially have gene flow from Linearis into its progeny, and it's not reciprocating. So there's a cost to hybridization. It's an uneven cost to hybridization in the production of F1 progeny. So what we were really interested in then is understanding how Linearis and Ratanii maintained their species differences despite being sympatric and co-occurring. And so we did a study in the greenhouse where we had 5 populations of Linearis and Ratanii that were sympatric, 5 allopatric Linearis, and 2 allopatric Ratanii. And we grew these plants to flowering in the greenhouse. And then what we did was at these different open flower stages, we would clip the stigma so that no self-pollination could occur. So these plants are capable of autonomous selfing, but if you clip the stigma, then at stage 1, it's no longer able to self-pollinate in later stages. And so by clipping the stigma of individual flowers at different stages, we were able to determine when self-pollination occurred, the stage of self-pollination occurred in each of the species. And we compared the sympatric populations of Linearis and Ratanii to the allopatric populations of Linearis and Ratanii. And what we found was, I think, really amazing and exciting. And it showed that when Linearis and Ratanii were allopatric, that is, when they were alone, they basically waited to self-pollinate when, our interpretation is they waited for pollinators to show up and self-pollinated late in floral lifespan. Whereas when Ratanii was sympatric, the black dots with Linearis, we see that it starts selfing much earlier and is selfing much differently from its allopatric populations. So Linearis remains delayed selfing, and Rutanii becomes more prior or competing selfing in David Lloyd's terms. So these data show that there can be a shift in the timing of selfing and the expression of mating system by imposed by the co-occurrence of a species that can cause the production of unfit hybrids in the population. We think this is really striking because when you think about populations having a range of outcrossing rates, as I showed earlier in the talk, it could explain why populations are moving back and forth between more selfing and more outcrossing, and also suggests a force that will drive populations that may be less selfing toward earlier selfing, basically becoming more and more high selfing because of the unfit hybrids in the presence of another species. So I hope that made sense. It's sort of funny to talk to you all in a void, but I'll just keep going. So another approach that we've taken in the evolution of mating systems and the role that selfing may play in adaptation and niche evolution is work that is done by my graduate student, Alani Grant, who just defended last month and is about to have a baby any day. So she's very interested in how mating system influences species distributions and what traits can facilitate, how self-fertilization can facilitate niche evolution. So when you think about niche evolution and you think about what happens when a species colonizes a novel habitat with respect to selfing, Sulphurs, when they move into a novel habitat as a single colonist, can have reproductive assurance because they don't require other individuals to be there to receive pollen. They can, in fact, autonomously self-pollinate. And selfing may act as a barrier to crossing with maladapted genotypes or other species, as I showed you in the first part of the talk, which can help retain the adaptations of the species that has moved into the novel habitat. And Mather, who's a geneticist from the 1940s, talked about selfing as freezing adapted genotypes. So if a good genotype makes it into a novel habitat, it can basically keep that genotype going, which is able to survive in the habitat that it has colonized. So if self-fertilization can enhance the likelihood of colonization and local adaptations, then we have two predictions: that sulphurs should have niches that are distinct from their outcrossing progenitors, and it's assumed that outcrossers evolved from sulphurs, and further that sulphurs will occupy wider niche breadths because they will have the capacity to colonize more new habitats and set up shop in a wider range of habitats than their outcrossing progenitors. So what we did was look at 14 species in 7 species pairs. So again, I told you that these are the species pairs, and we pruned the tree to make it obvious who we were using. And I just want to point out that Parviflora and Grandiflora are the species pairs that are found in northwestern Mexico. So what we did was, for all these 14 species, we collected 4,802 occurrence records from herbarium data, and we mapped those data to look at their distribution. So this is what they look like. This slide looks a little blurry to me, but the circles, the black dots, are the outcrossers, and the triangles, the gray dots, are the sulphurs. And we use these data, these distribution data, to do niche modeling, where we had the georeferenced species coordinate location data, and then we used data from databases on climate and others, altitude, other things like that, to calculate or to assign isothermality, mean temperature, annual precipitation, these kinds of climate variables to each of the locations. And then we ran Maxent species distribution models for each species to create maps of habitat suitability. So what is generated in this habitat suitability mapping is areas where we see that suitability is high in red or suitability is low in blue. And then from these data, we were able to estimate niche overlap, which is Schainer's D, and niche breadth, which is Levin's B. So here's an example of grandiflora and parviflora from North America. And you can see that grandiflora has a much smaller potential niche breadth or species distribution, projected species distribution, than does the sulphur. You can see it goes across North America, and it does. And this is what the maps look like in general. And the Outcrossers maps are across the top, and these are all blobs of North America. And the sulphurs are along the bottom. And visually, you can see that the habitat suitability scores encompass a much greater area for the sulphing species than for the outcrossing species. And in fact, when we calculate the actual niche breadth statistics, the niche breadth between the sulphurs and the outcross differs significantly between the species pairs, with the sulphurs having much broader niche breadths than the outcrossers, except in one case where, oops, where's my cursor, where, I mean, they're still significantly different, but in this case, Wrightii has a greater niche breadth than Torii. So that was pretty exciting. So it suggests that there's support for that, and that there's significant difference across all the species pairs when they're pooled, with sulphurs having much higher value of Levin's B, a significantly higher level of Levin's B than the outcrossers. And one of the interesting things is when we looked at the data that, sort of the climate data, we found that the sulphurs occupy drier niche space, that is like somewhat more harsh niche space than do the outcrossing species. And this is really interesting because a lot of people think about the fact that the ecological forces are about missing pollinators, but this suggests that this increased fitness under suboptimal conditions may be explaining why we see broader niche breadths for sulphurs, because they're able to persist in habitats that are more detrimental or more negative. And how might this work? So there are lots of hypotheses. Well, there are three main ones, but people have talked about this for a long time, and that is that sulphing in itself can alter water use efficiency. So in the first hypothesis, it's that small flowers relative to large flowers are limiting water stress for an individual. So in this case, sulphurs would just have a lower water use and a higher water use efficiency than outcrossers. Sulphurs may be selected to have a very rapid life cycle, and that is that sulphing evolves not because of benefits of sulphing, but the fact that being a sulfur means that the whole life cycle can go faster. And so being a sulfur means that basically how rapidly you go around the life cycle can be completed much more quickly than having to develop large and showy flowers and wait for a pollinator. And related to this, that short-lived flowers limit water stress, that if flowers themselves are not open very long, and because most flowers do not produce stomates and cannot control water loss from their petals, which is why you have to keep flowers, bouquets of flowers in a vase, that flowers that are small and selfing may have a shorter lifespan and basically limit water stress of plants. So together, these abiotic and indirect selective forces, along with the potential reproductive assurance, can explain why we're seeing such great niche breadth distributions for the selfing Collinsia relative to the outcrossing Collinsia. Okay, and so the last part of my talk is thinking about, well, how do you get to selfing if you're in a place with abundant pollinators? And can persistent pollinators create mutualism asymmetry? So what we're talking about is pollinators that continue to show up despite the genetic advantages of selfing. So again, when we think about these ecological forces, this biotic pollination area creates this whole group of species in the black here that are considered mixed mating. They make both a combination of selfed and outcross progeny. And so we are interested in my lab in melding the interaction between the ecological and genetic forces and understanding how they play out. So if you think about the transmission advantage, automatic transmission advantage, a sulfur makes offspring that have both of their gametes, plus it can contribute to gametes in outcrossed individuals. So this is the automatic transmission advantage, which should select for selfing. But with inbreeding depression, the fitness of the self progeny may be lower, and there's a loss or a low fitness of these self progeny. So despite the long-term interest in how these genetic forces act in populations, we still can't really fully explain why mixed mating is maintained and why selfing will be favored when inbreeding depression is less than 0.5. So when you look at the data from highly inbred populations up here, we see that I should explain this. So this is the x-axis are species with low selfing, mixed mating, or high selfing. And this is the measured inbreeding depression from experimental studies. What we see, this is the magic cutoff of inbreeding being 0.5. And what we see is that there is nobody that's high selfing with high inbreeding. I mean, if they're high selfing, inbreeding depression should be removed from the population. So what's going on in this place where we have relatively high inbreeding depression and mixed mating? It's widely accepted that reproductive assurance, which is we call RA, can favor selfing despite high levels of inbreeding depression. And reproductive assurance works when we have unreliable pollinators, conditions of outcross pollen limitation, or low mate availability, that result in selection favoring the production of self-progeny rather than no progeny at all. So to explain the presence of mixed mating with high inbreeding depression, we have to really think about where the ecology comes in, right? So when we look at the data for pollen limitation, we see that it's really not as strongly supported as you would expect. So here, these are fruit set, seeds per flower, seeds per fruit, or seeds per plant. You can see that there's really a very high category of low pollen limitation. And so the question is, why does mixed mating persist basically when there is this area here with low inbreeding depression? So first of all, we have to think about who they are. So most of these species are high to mixed mating species with low inbreeding depression. This group right here are mostly self-compatible annual or biennial plants. So they're plants that have a very rapid life cycle, and they can autonomously self-pollinate if their flowers are organized in the right way. So, so we can sort of gray out inbreeding depression and pollen limitation and think about pollen discounting and how that can work. So is pollen discounting really the driver? So we don't have really very many tests of pollen discounting, largely because they're really hard to do. And the data that we do have so far is pretty inconsistent. And so we're still stuck with this explanation, we need one, for why there are no to low inbreeding depression species with low or no pollen discounting, are mixed maters. It's just like, why do we have this? So when we think about it, what other ecological factors can erase the transmission advantage, this is sort of what we're left with, of sulphurs, having selfing and having this transmission advantage? Well, we are postulating, Rachel and I, that there may be a different kind of role for the evolution of selfing that explains these mysterious orange dots in this figure. So our hypothesis is that under the conditions that I've described, it would be beneficial for the plant to abandon the pollination mutualism because they would have a higher fitness if they could just self and then also do some outcrossing. But if pollinators are persistent and abundant, they may enforce outcrossing, which reduces the optimal selfing rate, and they can erase the transmission advantage. And that would slow down or severely, or stop, even halt it, the transition to selfing. So what we're talking is that the problem for the plant is that if the pollinators are moving pollen around, they're basically stopping the formation of self-progeny and producing more outcross progeny. So that's a problem for the plant. They can't evolve toward higher levels of selfing. So how can plants get away from persistent pollinators? Well, they can, we can think about this whole idea as a shift to autonomous selfing in a plant as abandoning the mutualism. And the progression of selfing can be slowed when self-compatible autonomous selfing plants are less reliant on pollinators for reproduction and fitness gains, but the pollinators still rely on the plant for the resources of pollen and nectar. So there's an asymmetry in the mutualism. So the plant's fitness would go up if it could self and have a transmission advantage, but the pollinator is enforcing outcrossing. So when we think about the evolution of selfing through this lens of mutualism breakdown, selection can shape organisms to maximize individual fitness, but this conflict, this asymmetry, conflict of interests are expected to arise when non-relatives interact. And the maintenance of the pollen mutualism is difficult to reconcile with natural selection favoring selfing traits that increase fitness of a plant partner. So this poses a challenge for the maintenance of the mutualism because each partner might benefit most from either exploiting or abandoning the other partner. So when might this mutualism break down for plant pollinators be constrained? So what we're framing is what we call the persistent pollinator hypothesis. And we ask, do certain ecological conditions exist where abundant pollinators remain reliant on plant resources and create this kind of asymmetry in the mutualism with continued outcross pollen delivery? And then what we're suggesting is that this asymmetry is likely to be the greatest under conditions where pollinators are abundant. That is, really little pollen limitation and very low conditions of pollen discounting. So that's what we think is going on in these together, the transmission advantage being wiped out by conditions where there are lots and lots of pollinators despite the transmission advantage. So what constrains evolution of selfing in species with low inbreeding depression? Well, when we look at pollination limitation, it's not really as pervasive as it has been chalked up to be. We find, as Knight et al. Say in their review, however, we find multiple lines of evidence indicating that the magnitude of pollen limitation may be overestimated. Specifically, publication bias, table 3, and fractional treatment, flowers, or reproductive episodes of plants led to elevated estimates of pollen limitation, as I showed you on the previous slide. Thus, it is clear that both publication bias and experimental methodology color our understanding of the magnitude and ultimately the prevalence of pollen limitation. So if you believe Knight et al., it may be that pollen limitation isn't that big a thing. It's maybe not a thing, and that this maintenance of mixed mating rather than the evolution of selfing is partly because of abundant pollinators. So if pollinator limitation may not be that pervasive, especially for self-compatible monocarpic plants, right? So these guys seem to make a lot of seeds and not have pollen limitation. It's low. And so this is a really complicated question and hard to answer, but to understand the evolution of complete selfing from mixed mating, we need to understand what traits might co-evolve with autonomous selfing, ability to enable self-compatible plants to escape their pollinators, and what factors would constrain them from being able to do so. So for pollinators, it's the rate of outcross pollen transfer, gynogamous selfing, facilitated selfing, pollinator abundance, and basically the pollinator population demography. How well and how abundant are these populations of pollinators? How reliable? And for plants, things that interact with the pollinator-driven factors are the floral display, flower size, shape, and rewards, whether it's self-compatible, whether it can autonomously self-pollinate, and when that autonomous selfing happens. Is it early in the floral development and can happen before pollinators show up to the flower, or delayed? So there can be escape routes for mutualism breakdown between plants and pollinators, that plants can self-fertilize earlier within a plant's lifespan. And here are some pictures of family level variance in flower size and the timing of selfing. So you can see that within populations, these guys might be able to escape. These small flowered early selfing individuals may be able to escape and self-pollinate and get away from these persistent pollinators. They could flower earlier in the season to create a mismatch with pollinators. However, there's a physiological limit for plants, and they can't grow too early or too late in the season because they just won't be able to physiologically manage those conditions. And it may be difficult to get away from these pollinators because the pollinators are also tracking environmental changes, and this sets up a red queen effect with persistent pollinators. You can reduce attractiveness in a plant. There can be selection for smaller flower sizes or less attraction, but that still may not be enough for persistent pollinators. And it's even been shown that flowers that are as boring and small as Arabidopsis are still visited by pollinators. So the data suggests that even the plant that we all think of as 100% selfing is not 100% selfing. So what we're trying to do now is do modeling and think about how these traits and conditions can come together to halt the evolution of selfing. What are the limits on floral traits that cause pollinators to abandon a flower? What are the levels of pollen limitation and pollen discounting for these species? Can we detect selection for traits that would allow plants to escape the mutualism when pollinators are abundant? And to what extent does this really happen? And I think that this all points to work that we're working on and others that recognize the importance of pollinators and species interactions of many different kinds in shaping plant evolution. And so it's not only the biotic conditions that deal with the pollination environment, but also the abiotic environment, as I showed with Alani's work, that are shaping the evolution of plant mating systems in this group. So just to kind of bring it to a close, this continuum of plant mating systems is a fascinating conundrum, and we're thinking about it all the time in my lab and thinking about how these processes evolve and how the genetic and ecological conditions interact. And we know the mating system is dynamic within a species, and in fact there's even a Collinsian here showing one of the mixed mating large flowered species, Collinsia heterophila. So there's a lot to be done. And one of the approaches that we're taking now is to try and look more into the genome and understand what genes are responsible or underlying the traits that we're observing that are part of the selfing syndrome or outcrossing syndrome within Collinsia. And just today we published a paper in Genes, Genomes, and Genetics that is just the first genome for the genus, and it has 7 chromosomes, and we were able to do some bioinformatics work looking at differences in floral gene expression in one selfing and outcrossing pair. So these are the 7 chromosomes showing the distribution of genes along the chromosomes, and the genes we've been identifying so far using Go analysis that relate to pollination, reproduction, development, and growth. So they're just color coded here. And we also looked fine scale at floral development in the very, very, very early bud stage. So we have 3 different buds of the sulphur Collinsia rotanii and Collinsia linearis, which is its sister outcrosser. And we are seeing that the species differ in pollen traits and in growth traits and reproductive traits in many cases, and the timing of when genes are turned on or whether they're turned on at all. So it's pretty exciting data that we have now, and it's opening up a whole world of possibilities for us to examine the parallel evolution of selfing and outcrossing across the entire genus and be able to ask the question if evolution answers the question of selfing the same way every time, or can different species evolve to selfing along different genomic pathways. So that is where I'm going to end the talk, and I am really happy to answer questions, and would love to talk to you after this. Okay, thank you.
Well, thank you. Thank you very much, Susan. Very interesting, as always. Very nice work you've done over the years. And we really appreciated your talk. So we will give some time for questions.
Is that all related?
What's that? Yes, there's a relay. But we know from the people that have been connected to our YouTube channel that there's people from Costa Rica and Argentina actually connected. So we have a broad audience. Okay, so
I know that some of these topics were covered in your class, right? Or related things in the class that you've been teaching, right? Yeah.
Yes, absolutely. Yes. And yeah, our students have, read some of your work, and yes, absolutely. So, well, I'll take advantage of of being the moderator, and I'll ask you a question. In the analysis the niche analysis that you did, where you showed a broader niche for the Sulphurs, right, than the Outcrossers. I wonder if you maybe this is something you're thinking about doing or did it. Did you look at the niche distribution of the pollinators of those of the Sulphurs and the Outcrossers, and does it overlap a lot? I assume that there's functional groups that will follow, especially the Outcrossers and the Sulphurs to a lesser extent, but do they overlap in distribution? Is it broader for the Sulphurs as well, or what? How does it look like, or you haven't done anything on that?
We have not looked at the pollinators at all. Okay. We have, and partly we haven't because they're the genus Collinsia is pollinated by generalist bees, so they're often a full range of small Andrinid all the way up to bombus and honeybees and all kinds of bees. So it's not like there's a strict pollination link with any particular set of pollinators. It would be interesting to look at the low absence of pollinators, but our data are suggesting that it's not necessarily the pollinator, but it's the climate. It's sort of the indirect selection for selfing that Sulphurs can move into niches that are more severe. Mm-hmm. But we did, as part of Alani's thesis, she also has done another analysis for selfing and outcrossing pairs outside of the genus Collinsia, and she's sampled across the angiosperms and has seen the same pattern that sulfurs are have wider niches and broader niche breadths than outcrossers. So I think it's quite interesting. Others who have looked at this question haven't found the same result, or haven't been able to find the same result, but they have mostly done just like polygon work where they draw a polygon around the distribution rather than understanding what is happening within that polygon. So
well, there's actually, we've been doing something like that actually with a graduate student of Silvana, Guillermo Huerta, who's probably in the audience, and found similar results. We did it for Ipomea, which is a genus, that we've been working with here in Mexico, and it follows a similar pattern to the one that you found.
but with
Between
So
Sulphurs and Outcrossers.
is that published yet?
No, it isn't. It isn't. It's actually, we did it for self-compatible and self-incompatible Ipomeas.
Yeah, we don't have
Yes.
outcrossing rates
Yes.
yet.
But
And the self-compatible species are pretty showy, they have pretty showy flowers. They don't have the selfing syndrome as clear as it is in Collinsia. So they may be more outcrosser, these mixed mating systems that have.
Yeah.
But
so did you use niche modeling or max extent modeling, or yeah? Okay.
Yeah. Well, yeah, my graduate student is about to finish that paper soon, so hopefully we'll have those results.
Interesting, because the people who have done the polygon methods aren't finding it, and the people who are doing it's harder to do the niche modeling, right? It's so much more time consuming to run all the.
yes.
Yeah.
Right.
But it seems like it's telling us a different story from the polygon method. It's exciting.
Yes. Well, going back to the pollinator thing, I was thinking that maybe, you do have several bees pollinating the Collinsia flowers, but I'm sure there's some that are more efficient than others, right? So what I was thinking was along those lines, if, for example, the distribution of the most efficient pollinator of the outcrossers is actually, more reduced as well, and following the distribution of those outcrossing species. That was more the thought that I had concerning that.
I wish I knew that. I mean, that would be a really interesting parallel study to this work, right? To be able to investigate who the pollinators are. And I mean, so Scott Armbruster has had some ideas about how pollen placement on the front or head or wherever of a pollinator may influence whether or not it's an efficient pollinator or not. We don't have those data. And it's hard, sort of depending on what scale of question you're asking, like having those kinds of data where you would know who the pollinators are across the entire range is like, makes my heart stop. But because you can imagine different pollinators might be more efficient in different habitats, right? And so even though this Anthena isn't there, this Osmia is really good at it over here. They're the efficient pollinators, but it looks like you don't have what you thought you needed. So you would really have to ground truth the biology of the pollination across the entire range, which that would be tough.
Yeah.
All right. I don't know, maybe
Thank
I
you.
should start reading the questions. I have a couple questions here. One is from Gerardo Avalos, and it says, it would be interesting to examine the evolution of selfing in introduced plants which find a new pollinator fauna, not adapted to the the evolution of selfing. Do you know of studies along those lines? And then he says, great talk, thanks.
Thank you. So I can, where do I start? Many species that are introduced to new areas that are successful invaders are often self-compatible and capable of autonomous selfing. But I know of some studies that looked at how pollinators have been pulled away from the native community onto the invader, and so that there's apparent lower levels of pollination going on in the native community relative to the invader being well visited. But I don't know of any like you're describing where a plant comes in and isn't visited and evolves to be more highly selfing. It seems kind of like that niche study that we did, that the colonists that show up and can self have high levels of selfing, and have high success as invaders. And I haven't seen the evolution of the mating system, but it would be worth looking to see if there has been a shift between the home range and the invaded range. Does that answer the question? Yeah, okay.
I guess so. Yeah. Thank you. And now we have a question by Jesus Alejandro Resendiz. And he says, for the niche model, this model account for the time axis? I mean, these niches are, wait, hold on a sec. Are they always on the same spot, or do they move as the climate variants occur? And what does it mean for global warming? Maybe we could ask, yeah, did you
I think I
get the question?
So we can only use data that are in the herbarium records, and we don't know places that may have been in the past and have died out, or places where it's just colonized. So we don't have a temporal sequence for any of the species. We did look temporally at the sort of the species age. So one thing I didn't mention in my talk is that the phylogeny that evolved and I and Scott published several years ago had a dated phylogeny. So we know when species evolved relative to each other. And we didn't see any kind of relationship between species age and niche breadth. So that wasn't your question, but that's a different kind of answer. So with climate change, I might expect the pattern to get even stronger, because if we're expecting more, the patterns of climate that are predicted in North America is that it's going to get hotter. Right? That means that we might see over long periods of time global warming. We may see the strong selection for more selfing occurring for those reasons. It's hard for me to share screen and see what's going on, but that one with the clock and the fast life cycle, all those reasons why selfing is selected for as a secondary trait rather than selection for selfing. right? So selection is a byproduct of selection for having a rapid life cycle, dealing with drought, dealing with all these other stresses. So it may mean that in the future, species that are selfing will do better than others. I hope not. I hope all the colonies stay there and like going, but you know, the worst case scenario would be something like that.
Okay, thanks. Thank you. We have another question by Guillermo Huerta, who is the student we were talking about, who works on hypomia. And he says, there is still this idea that selfing is an evolutionary dead end, which seems counterintuitive with a broader niche in sulfurs. Do you think there is enough evidence for the dead end hypothesis?
So there's a lot of evidence in the dead end hypothesis, and it mostly is based on data from incompatible species evolving to be self-compatible and then going extinct. So there has been less work in fully self-compatible genera, like the one I work on, like Collinsia. And I have talked to like Boris Ejik and Emma Goldberg and a lot of people that have made those models of selfing as an evolutionary dead end about this very problem. And it's, you know, we just don't have the magnitude of data on the fully self-compatible genera that we have on self-incompatible genera, self-incompatible evolving to self-compatible. But one of the things that I wonder about, and you know, if you think about the distribution of mating systems, it seems as if for some of these species, like the piece of the work that I talked about where they were sympatric and allopatric, that little tiny flowered Britanniae was outcrossing like a linearis when it was all by itself, right? So it suggests to me that selfing may be reversible under certain situations. And that's a kind of situation where you might expect it. And Stephen Wright and I wrote a grant proposal to try and look at the distribution of across many populations to see if we could detect that kind of reversal. But you know, they said they loved it and they didn't give us money, so I can't answer. It's really an interesting question, because I do, you know, I think the data support it, and seeing the magnitude of variation in so many species, that the likelihood that it's not just like going off one end and falling off. If it has low inbreeding, it's not going to go extinct. It's just not. Right. That's what I mean. That's what's supposed to be driving extinction is suppression. And so, if purging doesn't happen, then suppression will wipe out the sulfur. But purging works pretty well.
And then you have those persistent pollinators there.
Right. They never get to selfing.
Right. There's another question that's on a little different topic. So I will, well, just because this brings me back to the persistent pollinators, if I can ask my question now. This is about, I was wondering whether the persistent pollinator hypothesis would be more common or perhaps more appropriate for plants that are pollinated by pollen collectors rather than nectar feeders. Just thinking that you know, that's feels like selection to reduce pollen production may happen quickly, and it's easy to produce a flower that doesn't make any nectar, no? But you cannot make flowers that have no pollen. So the reward is always going to be there, whereas with nectar, you can just get rid of the nectar. So it would be interesting perhaps to look at that, whether this is happening.
really interesting. And I think, you know, a lot of these, so the species that are in that orange stack on that histogram were self-compatible, short-lived species. And a lot of those flower early, or, you know, rapid flowers, and maybe associated with pollen collecting bees, like spring emerging bees that just come in and feed their broods a collection of pollen. That's a really interesting thing. I'll mention that to Rachel.
All right. Okay, and let me see, there is another question in my WhatsApp chat. This is from Eric Fuchs from University of Costa Rica. And he's asking, are there differences in the types of genes between sulphurs and outcrossers? And he's referring mostly to if genes associated with morphology, or say genes associated with phenology or physiology, differ between sulphurs and outcrossers.
Well, so we are just in the very early stage. I mean, we literally published it today, and we're hopefully going to be able to answer that question. And I would love if others would use these resources to ask those questions. You know, there's so many questions that you can ask once you've developed some resources. You can get a little money to do sequencing, but to build the genome is like, it took us a long time to build the funds up. And so we do know, I don't know if I should open my talk up again. On the wrong direction. I can send the link to the paper. When I looked, they told us that we published it. I couldn't find the DOI, so you can look for it. Let's see. I'm trying to get back to the being able to share the screen, which I don't seem to be able to do.
You mentioned that there were differences in the timing for the expression of the genes, right, between the two groups.
Yes.
So that is probably affecting, that is likely to affect phenology, I suppose, flowering phenology, for example.
Well, it's hard to say. So can you see this?
Yes.
Yeah.
Okay. So, so to answer the questions, yes, we're seeing, this is Ratanii, and this is the distribution of the genes in Ratanii that we saw. So we know that there are genes in Ratanii that are influencing pollination, reproduction, development, and growth. And then when we did this transcriptomic comparison between Ratanii and Linears, just at this one stage. So we're not stepping out into really a large, we're not going very far out on a limb. Just saying that I know that for Linears and Ratanii, they have very different timing of expression or amount of expression between the sulphur and the outcrosser. So given that we were able to detect these kinds of differences just within the development of the flower itself, we think it's very likely that the kinds of things that you're asking, that how rapidly the physiology goes, or when it shifts from, okay, I'm done photosynthesizing for growth, I'm photosynthesizing only for seed production, or those kinds of decisions. I think they'll be very different. We know these sulphurs don't live as long as the outcrossers. They don't, you know, they wrap it up really quickly. Even their pollen has different longevity. So there are lots of phenotypic things that we know. We're just in the process now of getting to the being able to ask those kinds of questions. And I think if you are interested in that, I welcome you to contact us and think about a postdoc or or graduate school or something with Rachel or me or Stephen, because there are lots of things to ask.
Okay, thank you. Well, I think we don't have any more questions in the chat. There's comments. Thank you very much for the talk. It was very interesting. That's from Paulina Cruz and Alexis Rivera. Thank you very much. And I guess that's it. Do you have any more questions?
Well, I do. I have one more. The differences in size of the flowers you found, between the two groups, the Sulphurs and the Outcrossers. Are there also differences in the male component of the flowers? I mean, is the pollen of the Sulphurs smaller than the Outcrossers? And is also that related to maybe the male performance or not?
Yeah, I don't know if the pollen size is different, but certainly the bigger, I think, is less. And I think that's part of the reason why Britannia is not a good sire on linearis dam. It's it's the same as the the work that's been done in tomato, where sulfur can't outcross, and it's I forget the exact hypothesis, but it's basically there's not enough energy in those pollen grains to manage. Basically, yeah. So we do know that there are differences in the longevity of the pollen when we compare but yeah, I don't know, but I think that's it, and we've never done the measuring how far it gets down the style to determine whether or not that is the issue. There's not enough resources in the self-pollen to make it down the outcross style.
Right.
Yeah.
Well, yeah, you answered my other point, which was that might be explaining the asymmetry in the crosses. Yes. And very, very interesting. We find that some of that for some cucurbids actually that we are studying related to the sizes of different species of cucurbita that have been when we compare the domesticated with the wild cucurbita, we find differences in the flowers, but also those differences, we also find differences in the crossing ability between species, and that asymmetry is one that we're finding that is probably related to the pollen in itself, the pollen performance. But very interesting, yeah.
Very cool. Yeah, Donna Vogler found something too, right?
Yes.
Campanula. Yeah.
In Campanula, yeah.
Very cool. Very cool.
Well,
I mean, there's I feel like I've worked on this for my whole career and I still don't know so much. So always more science to do, which is why it's exciting.
Yes.
Yes.
Absolutely. Yeah. And thank you so much for sharing all the science with us. And it's
so great to see you both.
Yeah. Let's hope that we can have you here in Mexico sometime in the future.
Yeah. We should go and look for those populations in the north. Yeah. All right. Well, thank you very much. We really appreciate it. Thank you.
Bye.
Bye-bye.
Bye. Gracias a la audiencia.